Good morning, students. Today we'll discuss about WBC. WBC, they are known as white blood cells because of their color, and they are the real fighters and soldiers of the body. The WBCs or the leukocytes, they are the cells of immune system which are involved in defending our body against both infectious diseases as well as the foreign material. 5 different types of leukocytes they are present. They are formed from the bone marrow by a process known as leukopoiesis. They are formed from hemopoietic stem cells. The WBC, they live for about 3 to 4 days in average human body. And WBCs, they are found throughout the body. They are present in the blood as well as the lymphatic system. In this diagram, you can see the blue colored cells, bluish colored, they are the WBCs, whereas the pinkish colored cells, they are the RBCs. Now coming to the process known as leukopoiesis, it is the formation of WBC. During the formation of WBC, the stem cells, they are undergoing through various stages, that is promyelocytes, myelocytes, metamyelocytes, band cells and the WBC cells. So promyelocytes, their diameter is 15 to 25 micron and they are larger sized cell, rounded indented nucleus is present, and very important thing is they have numerous azurophilic, that is deep blue to purple, primary granules in their cytoplasm. Promyelocytes, they are responsible for the formation of granulocytes. We have 3 types of granulocytes, that is neutrophils, basophils and eosinophils. From the promyelocytes, myelocytes, they are formed. This is the first stage at which we can distinguish between these 3 types of the granulocytes because of the presence of the specific granules in them. The shape of the nucleus is variable, but it is again indented and displaced from the center, and the cytoplasm, it has azurophilic and specific granules. Most of the cell from this stage are neutrophilic. Since neutrophils, they are most numerous type of mature granulocytes. They form about 70% of total neutrophils. Then next stage is metamyelocytes. The diameter is 10 to 16 micron diameter, and there the cells, they can be neutrophilic, eosinophilic or basophilic, and they are bit smaller in size and they have indented and kidney shaped nucleus. Next form is the band form. Again the size is 10 to 15 microns. The cells in this stage have long thinned out nucleus with parallel sides. That is, it resembles thick bar stretched across the center of the cell, or more often horseshoe shaped. The rest of the cell still shows other characteristics of mature neutrophils. Then the band cells forms the mature cells, that is the segmented form. In this mature cells, the presence of several nuclear lobe connected by the filamentous strands of chromatin is seen, and the proper distinction between neutrophils, basophils and eosinophils is there. Now look at this figure. In this we can say this is promyelocytes with larger cells, rounded nucleus, and it changes into myelocytes. In the myelocytes, the granules, they become distinguished and they are stained differently with the different dyes. That is, acidophils, they are stained by acidic dyes, basophils, they are stained by basic dyes, and neutrophils by neutral. Then the myelocytes, they will be formed into the band cells. That is the eosinophils, they are the kidney shaped nucleus is there. Even in the case of the Neutrophilic myelocytes, the shape is again the indented nucleus is there, horseshoe shaped. So in the case of the band cell, almost the horseshoe shaped is there. But in the case of the segmented form or the mature cells, we can see that in the case of eosinophils, the horseshoe shape is there, whereas in the case of neutrophils, 3 to 5 lobed nucleus is present, whereas in the case of the basophil, S-shaped nucleus is present. So, leukopoiesis is a form of hematopoiesis in which WBCs are formed from the bone marrow. WBCs arise from the bone maturation of hematocytoblast, which are germinal stem cells for all blood cells and platelets, into various pluripotent stem cells, which can then further differentiate into different classes of WBC. So WBC basically they are of 5 types, that is granulocytes and agranulocytes. Granulocytes they are of 3 types, eosinophils, neutrophils and basophils, whereas agranulocytes they are of 2 types, that is the monocytes and lymphocytes. So the monocytes they develop from promonocytes and the lymphocytes they develop from pro-lymphocytes in a separate line. Now coming to the granulocytes one by one. All cells in this category contain secondary granules and they have the multi-lobed nucleus. That is why they are also known as polymorphonuclear leukocytes, that is PMN. A very important question from examination point of view. What are PMN? They are having multi-lobed nucleus, that is neutrophils. 70% of total nucleoside, total WBCs or leukocytes, they are formed by the neutrophils. They are most numerous, so they are easy to identify. Their cytoplasm is pink to gray because of neutral staining of specific granules. So neutrophil function as scavengers within the extravascular tissue, destroying bacteria and other infectious organisms that invades the body. The neutrophils, they contain 3 types of granules. Number 1, azerophilic granules or the primary granules. They are formed by myeloperoxidase, bactericidal increasing protein, defensins, and the serine proteases, neutrophil elastase, and cathepsin G. Whereas the secondary granules, they are formed by alkaline phosphatase, lysozyme, NADPH oxidase, collagenase, lactoferrin, and cathelicidin. The tertiary granules, they are formed by cathepsin and gelatinase. So neutrophil release these proteins in 3 types of granules by a process called degranulation. So degranulation is release of these granules from their cytoplasm to outside to perform the defensive function. So neutrophils, they show hypersegmentation in B12 and folate deficiency, very important. And very important thing is the neutrophils of male and female organism is different. The cell nucleus of neutrophil from a female having additional X chromosome shows neutrophil drumstick. Drumstick is in this first figure, you can say the protuberance from the one lobe of nucleus which is there, which is known as the drumstick. So male and female blood can be identified through the presence of drumstick. The female has the drumstick, but males they are not. The nucleolus disappears as the neutrophils mature, which is sometimes that happens in only few other types of nucleated cells. In the cytoplasm, Golgi apparatus is small, mitochondrial ribosomes they are sparse, and rough endoplasmic reticulum is absent, and about 200 granules they are present in the cytoplasm. When circulated in the bloodstream and unactivated neutrophils, they are spherical, but when activated they change their shape, they become amorphous, they become amoeba-like and extend pseudopods as they hunt for the antigens. Next is the eosinophils. Eosinophils constitute 2 to 4% of the total leukocytes. These cells usually contain bilobate or horseshoe-shaped nucleus, and their cytoplasm is full of brightly stained eosinophilic, that is orange-red specific granules. The eosinophil function specifically as phagocytes to destroy the larvae of parasites that have invaded tissues that like trichinosis, schistosomiasis, and appear to play role in allergic response. So whenever helminthes infection is there, the amount of eosinophils they increases in the blood. The other function of eosinophils include phagocytosis of antigen-antibody complex because they have to be removed from the blood. Next next cells is the basophils. The basophil they constitute about 0.5 to 1% of leukocytes, and they are more difficult to find in the routine blood smear. Their nucleus is S-shaped. The cytoplasm is full of dark purple specific granules. The basophils, they are phagocytic but function like the mast cell. Their granules contain histamine and heparin, which play important role in initiation of acute inflammatory response. So my sincere thanks to www.google.com for providing us beautiful pics so that we can explain our topic well.